Good morning students. Today we discuss about bacterial growth curve. First, what is bacterial growth curve? So bacteria is a unicellular organisms. It is a growth curve means bacteria is introduced into a liquid growth medium or solid culture media. The population is counted at intervals. It is called as bacterial growth curve. It is possible to plot a bacterial growth curve that shows the growth of cells over time. Introductions. Bacteria are unicellular organisms. It reproduce asexually. That means for binary fission. So one cell divides into 2 and then 4, then 8, 16, 32 and so on. It is called binary fission of bacterial reproduction. That image shows that the binary fission of bacterial reproduction. Bacteria is a unicellular organisms. The time taken for a bacterial cell to double is called generation time. The generation time varies among different species of bacteria based on the environmental conditions and they grow in. Clostridium perfringens is the fastest growing bacteria and the slowest bacterial growth is Escherichia coli. Mycobacterium tuberculosis is the one of the slowest growing bacteria, taking about 12 to 16 hours to double. So growth curve, it is one of the closed system. It contains for enough nutrients and bacteria shows a predictable growth pattern, that is the bacterial growth curve. It consists of 4 different phases. First one lag phase, stationary phase and death phase. This image also shows the bacterial growth curve plot. First one, lag phase. So the bacteria introduce the new environment. The beginning of multiplication is known as lag phase. In this phase, the bacteria does not reproduce but prepare itself for reproductions. The cells are active metabolically and keep increasing in size. The cells synthesize RNA, growth factors and other molecules required for cell division. Second phase is log phase. It's otherwise called as exponential phase. This phase is marked by doubling of the bacterial cells. The cell number increases in a logarithmic fashion such that the cell constitute is maintained. The log phase continues until there is a depletion of nutrients in the setup. It is one of the rapid exponential cell growth of the log phase. Third phase is stationary phase. After log phase, the bacterial growth almost stopped because the lack of essential nutrients and lack of water, oxygen molecules, changes in pH of the medium, etc. and also accumulation of their own toxic metabolic wastes. It's during this phase that the culture generates population density. So it is a death rate of bacteria is exceeded. So it also produces the endospores during this stage. Bacteria become gram variable, so irregular staining. Many bacteria start producing this phase for exotoxins. Fourth phase is death phase or otherwise called as decline phase. This phase all bacteria leads to death of the cells. So because it is a lack of nutrients, physical conditions or other injuries to the cells. That's all about the bacterial growth curve. Thank you students.